Welcome to Unit 5 of Module 2. As this is our kind of sciencey module, we've just looked at kind of the evolution of mummy science as a discipline. And now this is something that I know you've covered. It's called the scientific method. It's something you've covered in a variety of coursework in your past. And in actuality, the scientific method is really not just a part of the scientific community. It's simply a ordered way of logical thinking. And you can use, and you probably do use, the scientific method in many of the decisions that you make or anything that you want to learn or find out. So we're going to look at the scientific method. We're not going to spend a lot of time on it. I think you've been exposed to this many times in the past. So we're going to get ourselves through the scientific method, but I just want to make sure that we are all on the same page. You've probably seen a model something like this, the scientific method. First, what's the question? Now, prior to the question, typically science begins, as does your curiosity, begins with an observation. You see something. You wonder why something is the way it is, or you wonder if X relates to Y, or whatever the question might be. So once we've made an observation, we have to formulate a good question. So the question becomes, what do you want to learn about? What is your question? We're going to go through an example in the framework of mummy science in a moment so you can put the scientific method in order. Then we conduct some research. And by research, we're talking about background research. You gather information. You find out what else is known about the subject or about the question or the variables within that question. Next, you develop a hypothesis, an educated guess of an answer to your question. If I do this, this will happen. It's an if-then statement. And there's something called a null hypothesis too, which we'll address momentarily. Then you develop your procedures and methods. How are you going to experiment? You're going to design your experiment. How are you going to answer your question, test your hypothesis, and answer your question? And you build that procedure, those procedures and methods based upon the background and research that you've discovered in your information gathering session. Once you've conducted the experiment, you collect the data. Now, the data is what you collect during the information. And then you make your observations. So they're the written description of what was noticed during the experiment. So you start with your question, you see what's been done, you formulate hypotheses, you devise and conduct an experiment and collect the data, report the data, and make your observations about those data. In other words, a description. And most papers end with a written discussion or conclusion. So now you make a conclusion based on the outcome of your experiment. Was the hypothesis correct? Now recall from the last presentation in Unit 4, we looked at basic study designs in mummy science. And first of all, there are case studies, as you know, in which these are individual studies, but these lead to an interpretive analysis in which we gather information. And there are many experimental designs. We notice something unusual about that individual. We come up with ways to examine that aspect of that mummy or that individual. And then we collect that data, make our observations, and make our conclusions. And remember, a case study adds to it. It's like a small volume or chapter within a book. Population studies are the whole book. Experimental designs are larger. Now that we know how to find that disease or bony structure or anomaly in one individual, can we find it in hundreds of individuals? And then can we compare the men to the women, the adults to the children, so we can get some groups analysis within that population. We can compare population X to population Y. Well, we can get some strong statistical analyses and conduct these statistical procedures, and determine the degree to which this happens, and then we're finally being able to create a knowledge base, something that we can positively say about this population. 34%, for example, had this disorder. All right, now, remember, all mummy surveillance begins with proper background. We need to know what's happening behind us once we've made an observation. And here's one of my favorite sources and references for background information in mummy surveillance. The Weekly World News. Alien mummy goes on rampage. So now we've got to investigate. We've got to go out and see if we can find this alien mummy. Just kidding. Of course, your background has to be legitimate. It has to be from scientific literature. It can be anecdotal reports, because anecdotal reports are important as well. They help perhaps support the question or the observation and the question that you've come up with. All right, so let's go through a study using a mummy. First, we develop a question. Here's our research question. This is hypothetical. Was ancient population X afflicted with intestinal parasites? All right, now we conduct background research. We look for articles and reports of other cultures who had intestinal parasites. We learn from those reports the methods that they used in their studies, so we can reproduce those methods in our study. So we use those methods for our study design. That's what we're trying to get out of the background research. And then we also try and discover what is already known about population X. What was their diet? What was their culture? What did they eat? What were their implements? Did they fish? Did they hunt game? Or were they primarily vegetarians? Now, once we have our background completed, we establish a hypothesis and a null hypothesis. Our hypothesis from our study, based on our background and related to our question, is if we study coprolites, that's the ancient poo, from population X, we will find evidence of intestinal parasitic infection. But we also in science develop a null hypothesis. If we just went with a hypothesis, it looks like we're trying to prove something. And what's true about science and what's true about any investigation you can think about in any of your disciplines, remember, this is just a logical way of thinking. We want to remove any bias. So we develop what's called a null hypothesis to try and control that bias. So the null hypothesis is, if we study coprolites from population X, we will not find evidence of intestinal parasitic infection. In other words, there's not a relationship between parasitic infections and our population, when we're speculating that there may be. But we have to study it if there isn't in order to discover it. Now we establish our procedures and methods. And remember, this is based on the research, on the background information that you've been able to gather. First, we're going to collect coprolites from the mummified remains of population X using aseptic technique. Second, we're going to record the data collection locations and demographics of the populations, and anything that we want to relate the results of the study to. We're going to rehydrate and stain the coprolite samples. We're going to conduct microscopic identification of parasitic ova using XYZ parasitic ova identification methods. We're going to record and report the frequency, if any, as a function of the overall population and as a function of any of the demographic data that we may have collected. So now we have our experimental design. We're going to collect it. We're going to record information. We're going to rehydrate and prepare the coprolite samples, and then we're going to examine it for parasite ova. Then we're going to record the frequency. This coprolite came from mummy number X37. So we're going to mark X37 on the coprolite sample, and X37 did or did not have parasite ova. It had this type of ova or worm or whatever it might have been, and it was a male. So now we can begin to compare our data later on. It makes perfect sense. Now we have our data. That's an assessment and recording of the actual number of type of parasite ova present in the coprolite samples from population X, once again broken out by demographic data. We make our observations about the data. Now this represents the interpretation of the data. In our sample, there was an overall, this is all hypothetical, there was an overall 35% infestation rate among the sample population. Hookworm was the most prevalent parasite identified. Demographic distribution of the ova was unremarkable. So that's our interpretation of the data. Now, beyond that, we pass on to conclusions. And our conclusions, we try to answer the question, what does it all mean? In our conclusion, we state that we have proven the null hypothesis to be false. Remember, in the null hypothesis, we said there would be no parasites in population X. But we did prove the hypothesis. There is a relationship between parasite ova and population X. So, in our conclusions, population X was infected with intestinal parasites, and we can state which specific parasites they were, ... And the demographics had no impact on the distribution. And what does that mean? Well, that means probably they were all eating the same thing, for example. Now, what's really important about the scientific method in mummy studies or in anything is that once you've come to a conclusion, you've done your Remember, it's funny, the scientific method started with observation and your question. It came all the way down to data and observation, which is your interpretation of the question. And then in your conclusions, you end up with discussing the observations of the data. And generally, what happens in this just before the conclusion here, what happens in the observation and your discussion of the data is that more questions arise. And that's what the scientific method is intended to do, kind of complete a whole circle. And as we roll that circle and that ball of the scientific method over and over, slowly and slowly we learn more. But remember, good science begets new questions. Science never really proves anything. And that's one of the problems with that a lot of folks have with science or quote unquote scientists is that they're a little sometimes aloof and act like they know everything. But true scientists know that we know nothing. We only know infinitesimal small parts of what there is to know. And that's an important aspect of science. We're learning, yes, and what we do learn, we learn accurately. As we'll learn later in this course, there can be a lot of bias in science, in mummy science and other sciences and other disciplines as well. All right, that's the end of Unit 5. I wanted to make sure we were all on the same page with the scientific method. Let's move on to Unit 6, in which we're going to complete our Module 2, kind of this science-y background area, and we're going to look at what's supposed to happen when you die. Decomposition. Not mummification, but decomposition. We have to understand decomposition so that we can understand how decomposition is stopped and how mummies are made. Great, we'll see you in Unit 6.